Hello everyone and welcome to another MCAT ACE lecture. So this lecture will be focusing on the genetics and evolution concepts that are being covered in the MCAT. We will divide this video into two parts. The first part, we will go over the Mendelian concepts and then go over some of the factors that affect genetic variability. And then in the second part of this lecture, we will continue the factors that affect genetic variability and then go over some of the analytic methods as well as the processes of evolution. Okay, so starting with the Mendel's genetic experiment. And as you all know, Mendel is known as father of genetics because the modern science of genetics was founded actually by Gregor Mendel, and he was an Austrian monk that was in charge of the monastery garden. And it was right there that he has started to carry out his experiments with the ordinary garden pea plants, which has led to the discovery of the law of segregation, which I'm going to cover right here. Mendel's first law was law of segregation, as well as the law of independent assortment. Now, the reason that these pea plants were excellent for genetic experiment was that they had a small size, they were easy to grow, and they were yielding many different progeny. So that was some of the advantages of this model system that Gregor Mendel has been working with. And the pea plants that he was working with were purebred, meaning that they were producing identical offspring to themselves. So what Mendel did was that he started a cross between two purebred plants, one with a purple color and another with the white color, and he noticed the first generation, which was hybrid because it was a combination of two purebred plants, the first generation has yielded only one of the colors of the two he has started the experiment with. So the first generation was called F1 generation, and these were hybrid plants. The second generation was another cross. It was self-pollination between F1 and F1. So we have uppercase P and lowercase p. It was crossed to another sibling. So it was crossed to another uppercase P, lowercase p. So the pollens that are being generated by each one of these plants is, for the first one, we have uppercase P, and for the second part of the genotype, we have lowercase p. Same for the other plant. We have an uppercase P and lowercase p, and consequently the crossing of these pollens will give rise to capital P, which has a purple color, capital P, lowercase p, which has a purple color, again capital P, lowercase p, that has a purple color, and then finally lowercase p, and since this is lowercase p is a recessive gene, now this plant was able to express the original phenotype of its parents. Okay, and so the ratio here is 3:1. 3 purple colored flowers in comparison to 1 white flower. So these experiments has led Mendel to come to two conclusions. One was Mendel's first law, which was law of segregation, stating that during meiosis, alleles will segregate from each other so that each gamete carries only a single copy of each gene. So here you can see that each gamete is only carrying a single copy. It can't have two P's. So each gamete is only carrying a single copy of each gene. So based on law of segregation, during meiosis, alleles will segregate from each other so that each gamete will carry a single copy of each gene. And then we have the law of independent assortment, which states that the different pairs of alleles will segregate independent of each other during meiosis, provided that they are not linked together, meaning that they are not on the same chromosome. So alleles will segregate independent of each other, provided that they are not on the same chromosome. And this law is referred to as law of independent assortment. Okay, and so let me here give you an example of law of independent assortment. Here we have an allele for the shape of the P, which is shown by A, and we have another allele for the color of the P, shown with the letter B. These alleles will segregate independent of each other. It's not that A will affect B, as long as they're on different chromosomes. Okay, so the very first examples that I've shown you was for a single trait, which was the color of these P's. Here we have a dihybrid cross because we are going over two different phenotypes. One is the shape of the P and another is the color of the P. And here I have shown you that the capital letter B is a dominant gene for a round shape of the P, while capital letter A is a dominant trait for yellow color. On the other hand, we have the recessive traits, which is shown as lowercase b. So two lowercase b's will yield wrinkled p's, while two lowercase a's will yield green color. Okay, so this would be a good practice for you if you could try to solve this question and let me know what would be the proportion of different phenotypes in the F2 generation. So just remember here, AABB has been self-pollinated. So this has been crossed to AABB. So now you can go ahead and pause the video, solve this question. Once you have the ratio of phenotypes in the F2 generation, you can replay the video. Okay, I hope you had enough time to solve this question. So let's show the type of pollens and ovules that we will get during this cross. So for the very first one, we have a capital A and capital B. That would be the first combination. So A, B. Then the next combination that we can have is capital A with the other gene and lowercase b. So we have A, B. Now let's move on to the next gene. Here is the lowercase a. The first combination is lowercase a going with the very first one, capital B. So we have A, B. And then the second combination is again lowercase a going with the second genotype, which is lowercase b. So we have lowercase a, lowercase b. Now same thing exactly happens right here. If you repeat that for the ovules, we will get capital A going with capital B, so AB. Then we have A going with the lowercase b, so AB. And then the next combinations would be A going with B, so we have lowercase a, uppercase b, and then lowercase a going with lowercase b, so A, B. And if you solve for this question, if you put everything together, so here we have uppercase A, uppercase A, uppercase B. With this combination here, we have uppercase A's and one uppercase B, one lowercase b, and you keep filling this box. This is what you will get here. So I'm going to erase this box here and show you already the solved question. Okay, so that would be the combination that you will get, and I've already shown you the type of phenotypes that every one of these genotypes will yield. So the answer is that you will get a 9:3 to 3:1 ratio. Okay, and this one is the green colored P with a wrinkled shape. Okay, similar to the parental recessive trait that has been shown there. Okay. So now let's go over some definitions for the Mendelian concepts. The first one is locus. This is referred to as location of a gene on a chromosome. So if you have a chromosome and this is the location of the gene for the color of the P, for instance, that would be the locus. Then we have genotype, which is the genetic material on a given locus. Like for instance here, we are having genetic material of AABB, or uppercase A, uppercase A, uppercase B, uppercase B. So that would be the genetic material referred to as genotype, while what the genotype manifests as, like for instance round P with a yellow color, that would be referred to as phenotype. So genotype is the genetic material at a gene locus, versus phenotype is the manifestation or external expression of these genes, like for instance color of the skin or the blood type. The next term is called pleiotropy, and that's when a single gene will lead to expression of 2 or more phenotypes. And I'm going to give you an example. The gene that expresses melanin, for instance, if that is defected, then it would lead to albinism, where multiple organs will be affected. Like, for instance, these people will have a very fair skin, they will have a white hair, and they would have a pink color of the iris because there is no melanin in these tissues. Then we have the allele, which is a variant of a gene. For instance, here we had the allele for capital A versus lowercase a, because it was a variant. One was color for yellow, the other one was color for green. Or capital B versus lowercase b. So these are variant of a gene and referred to as allele. Here I've given you some examples. White versus red flower. These are like examples of an allele. Or blood type A versus blood type B. Both of them are the same gene. They are both focusing on the blood type. Same function, I should say, same function gene, but they're different from each other the way they express. Then we have the definition of homozygous and heterozygous. Homozygous is when the alleles at a given locus are the same, like for instance lowercase a, or uppercase A. Versus heterozygous is when the genotype consists of 2 different alleles. So the example given here, this plant and this plant, both of them that are purebred are homozygous, versus their F1 generation is heterozygous. You see we have capital A and lowercase a, and then capital B and lowercase b. So example of heterozygous is right there. Wild type is basically referred to a normal allele or phenotype of an organism. So like for instance, the flies that you find outside in the world, those are wild type flies, versus those flies that you mutate in the lab, those are no longer considered wild type. Okay, so wild type is a normal allele or phenotype of an organism. Recessiveness is referring to genes that are only expressed when they are present in an homozygous individual, like lowercase a, it can only get expressed when it's in a homozygous state. Once it combines with a dominant gene, it can no longer express itself. Like for instance, capital A is the dominant gene and will express itself, even if the recessive gene is present. So here we have a heterozygous A, capital A, lowercase a, but the recessive gene will not be able to express itself. So here, recessive alleles of a gene are denoted by a lowercase letter, while dominant alleles are denoted by an uppercase. And all these examples that I've solved here for you were examples of dominant versus recessive. Here, the white color of the flower was recessive, while the purple color was dominant, and that's why in the hybrid cross only the purple color has shown up. Same happens with the examples I've given you here. The round color and the yellow color were dominant, that we're here demonstrating in the hybrid again, versus the wrinkle shape and green color of the pea, those were recessive phenotypes. Okay, codominance is when both alleles contribute to a phenotype. Like for instance, we have blood type A and blood type B because of the type of genotypes that they're being expressed on the surface of these red blood cells. Once you cross blood type A with blood type B, you would get blood type AB. And that is because both of these antigens are now being expressed. So that's why it's referred to as codominance, because both alleles contribute to phenotype. On the other hand, we can have incomplete dominance, where neither one of the allele can take over the other one. And so consequently, the heterozygous individual will exhibit a phenotype that is in between the 2 parental traits. Like for instance, if you cross a white snapdragon flower with a red one, this cross would yield pink flowers, and that would be an example of incomplete dominance. Leakage refers to gene flow from one species to another. Penetrance is the percentage of phenotype that will express their related genotype. Like for instance, when I say that retinoblastoma has 90% penetrance, it means only 90% of patients who have mutation in their retinoblastoma gene will show cancer, 10% will not. So that is referred to as penetrance, basically the frequency that the genotype will manifest in the phenotype. Or for instance, you say that BRCA1 gene mutation has an incomplete penetrance, meaning that not necessarily all the individuals who have this mutation will develop ovarian or breast cancer. On the other hand, we have the term expressivity, and basically that refers to how much of the phenotype is actually being expressed in relation to a genotype. Would it be a very severe phenotype that would be present, or a very mild phenotype? And this example is shown in patients with neurofibromatosis type 1. They all have the same genotype, but some individuals show a very severe form of the disease versus some individuals show a mild form of the disease. Again, they have the same genotype, and that is due to variable expressivity. Now, I would like to stress out that with variable expressivity, these patients still will show some phenotype, even if it's mild. Versus penetrance, it's possible that the individual may not show the phenotype at all because there is incomplete penetrance. Some of them will not show the phenotype. The next terminology is hybridization, which we've already discussed. Basically, that's the process of interbreeding individuals with a purebred genotype, and that would yield a hybrid population. And then finally, gene pool refers to all the alleles that are present in a population. That refers to as our gene pool. Okay, now let's go over the process of meiosis and discuss how this process can lead to different genetic variability. So far we have gone over the Mendelian concepts. From here on, we would like to go over factors that can affect genetic variability, starting with meiosis. So in our separate reproduction video, we've discussed extensively how germline cells that are 2N can undergo meiosis to yield 1N, which are either sperm or egg. It could be 4 sperms or 1 egg plus 3 polar bodies. If you don't recall the content, you can refer always to our reproduction video. But then, unlike mitosis, where 2N yields 2N, one other difference in meiosis is that these chromosomes during prophase of meiosis 1, so during prophase of meiosis 1, homologous chromosomes will undergo recombination or crossover, and consequently there would be genetic exchange between chromosomes. This process will lead to variability in the type of sperms and eggs that are being produced. Okay, so here let me show you an example. We have one homologous chromosome right here, and then another one that comes over, and here a crossover happens where there would be genetic exchange. Consequently, the cells that arise from meiosis would not be identical. These 4 cells can have different genetic material. Now let's go over the mechanism of crossover. So basically there are 3 unique features in meiosis that are different from mitosis, and that is during recombination there would be a synapses formation where there would be pairing of homologous chromosomes along their length. So here, as I've just shown you, we have synapses. And then in the next step, there would be crossover between the chromosomes in order to allow genetic exchange, and that happens at a point that is called chiasma. So here I have provided you with a nice drawing that shows you here the homologous chromosomes has been lined up, and these pairs of homologous chromosomes are referred to as tetrads. And then the point that the 2 chromosomes cross over, or in other words form an X-shaped chromosomes, this is referred to as chiasma. And you can see that as a consequence of this crossover, now we have some genetic exchange between the chromosomes. Now in the meiosis 1, these 2 homologous chromosomes will separate from each other. So let me show you. So this is what happens in meiosis 1. The 2 homologous chromosomes are now separated from each other. And now in meiosis 2, the sister chromatids will separate from each other. So this sister chromatid will separate from that one, and here this sister chromatid will separate from that one. And so you can see that the products of what we got here, and so these will be incorporated into different sperms, for instance. So we have 4 different sperms that are haploid, and their genetic content is different. So by having recombination, meiosis will yield genetic variability in the offspring. And then finally, the other important difference between meiosis and mitosis is that reduction in the chromosome number. Meiosis yields haploid gametes, while mitosis yields diploid. So here we have 2N, and mitosis will yield 2 2N cells. So this is mitosis. Versus meiosis, we have a 2N, and it undergoes 2 division, meiosis 1 and meiosis 2, as a consequence of which it will yield 4 haploid cells. So these 4 could be sperm, or if you are talking about female, because there is disproportional distribution of cytoplasm, 3 of these gametes will have very little cytoplasm. So these are called polar bodies that would degenerate, and only one of the eggs will survive. Now moving on with the next concept, which is linkage, crossover, and genetic mapping. So basically, based on the frequency of crossovers, we can tell how close 2 genes are located to each other. So here we have 1 gene here and another one here. There is higher chance of crossover during recombination between these 2 genes versus 2 other genes that are located very close to each other. So here there would be lower frequency of recombinant chromosomes versus the green one will have higher frequency. And so based on the ratio of crossovers, we would be able to create a genetic map for different genes, and I'm going to go over that in a few minutes with you. But first I would like to discuss how Mendel's law of independent assortment was stating that during gamete formation, segregation of one gene pair is independent of the other one. But this was only for traits that are located on different chromosomes. And here, as I've shown you in this example, for instance, A and B, these 2 genes are being independently assorted from each other to yield these pollens and ovules. And that is due to the fact that they're being located on different chromosomes. Now, what happens if they are on the same chromosome? If they're on the same chromosome, then there is more likely that 2 genes will be segregated together. Because think of it, here we have a chromosome. And if we have 2 genes that are being located very close to each other, these 2 genes will happen to be together most of the time, unless we have a crossover right in between that separates these 2 genes. And so that's why we say that the law of independent assortment of Mendel's is only valid for genes that are on different chromosomes. And it was actually Thomas Morgan that has first come with the concept of linked genes. So linked genes are referring to genes that basically seem to violate the principle of independent segregation, because these genes are located on the same chromosome. And so the 2 conclusions that Thomas Morgan has came to by studying Drosophila melanogaster, which is the other name for the fruit fly, it was that number 1, genes on each chromosome are genetically linked. And then his second conclusion was that Mendel's principle of independent assortment only applies to chromosomes. So independent assortment law of Mendel applies to chromosomes. Okay, not genes that are located on the same chromosome. So what happens if there is no crossover? Under those circumstances, the 2 genes that are on the same chromosome will segregate together and will inherit to the next generation. However, if there is crossover between those two genes, then they would separate from each other and present on two separate gametes. And so the chance of the crossover, as we just discussed, between two genes increases as the distance between those two genes increase. And so here I'm going to show you an example on how we can use the recombination frequency in order to find the genetic map or the genetic distance between these genes. And so what you have to do is that you have to find out how many crossovers happen between M and R. So the potential crossovers that we can have here is we can have a crossover between M and H. Okay, and so here is an example of the gametes that results from crossover between M and H. Alternatively, we could have a crossover between H and R. And under this circumstance, these would be the type of gametes that we get from crossover between H and R. Alternatively, we can have two crossovers between M and R. Okay, and here these are shown as M plus R and plus H plus gametes. Okay, for this one, however, since two crossovers has happened between these two genes, you have to multiply the value by 2. And the formula that is shown here shows you how to calculate the recombination frequency between M and R. It's basically the value of single recombinants, which are shown right here, plus the number 2 times double recombinants, because these double recombinants have two crossovers that had happened between M and R. Okay? And you would actually better understand this question once I give you an example. So here it's giving you, there is a practice question that tells you the fruit fly genes W, X, and Y are all located on the same chromosome. The following table depicts the number of gametes that are produced in a female fly with the genotype +++ and genotype WXY. So these are our parental genotypes. What is the distance in map units between genes Y, W, and X? So the very first thing that you need to do for this example is to find out the order of genes. Which two genes are most farther apart and which one is located in between. This is actually easy to answer, because what you have to do is that you have to look for the two gametes that have the lowest frequency. And in this example, plus W plus and Y plus X have the lowest frequency, meaning that these two gametes have developed from a double crossover, because double crossovers are so rare that the gametes that result from double crossover have the lowest frequency. So therefore, given that these two are the result of the double crossover, therefore the gene W must be located right in between. So here we are having YWX genotype in the parents, and then another chromosome has +++. Now what happens if you're having a double crossover? So we are having one crossover here and another crossover here. Follow the path of crossover in order to find out what gametes you'll get here. So we get a plus W plus. So plus W plus is one gamete that we get from this crossover. And then for the other side of this chromosome, we get Y plus X. Y plus X. All right, so we've answered the very first question here. We know that the order of these chromosomes is W being located in between. So we have Y, W, X, or it doesn't matter. You can write it X, W, Y. The important point is that the gene Y is located in between. Now we can move on with the next question, that is to find the distance in map units between genes Y, W, and X. Alright, so the very first thing that we would like to do here is to find the distance between Y and X, which are the two genes that are farther apart. So which frequencies did we have to add? Do you recall? Map unit was equal to something divided by total. It was the single recombinants plus any double recombinants that had happened times 2. So the very single recombinants that we can have here is one crossover here. It would give you plus WX, so plus WX, or the other side of the chromosomes gives you Y plus plus, Y plus plus. So these are from recombination between Y and W. We could also have recombination between W and X. So the result here would be YW plus YW plus, or it would be plus plus X. So the genotypes that are shown right here, these are all single recombinants. But then the gene Y and X are so far apart that there is even a chance of having double recombination. And so those double recombinants we just discussed, we have plus W plus and we have Y plus X. Okay, so here those genotypes are shown right here. But since the two crossovers has happened, we have to multiply this number by 2. 2 times Y plus X frequency and plus W plus frequency. Okay, I hope it makes sense. Once again, between Y and X, since 2 crossovers has happened here, we have to multiply this value by 2. If we were talking about what the distance between Y and W, these double frequencies, there is only one that has happened between Y and W. Or if we were talking about W and X, there's again only one that happened between W and X. So we didn't have to multiply this value by 2. But now in this situation, for Y and X, 2 has happened between these two genes. So we have to multiply this number by 2, whatever it is. So let's go back to our table and find out what numbers we have to add up. So again, this was the formula that we had to use for finding the distance between chromosomes X and Y. Now referring to this table, let's find out what each one of these are adding up to. So the very first 4 are single recombinant, while the last 2 are double recombinant, and we just figured that out at the beginning of this lecture. Okay, so how many Y plus plus gametes do we have? We have 12 of that. How many plus WX we have? We have 16 of that. How many YW plus we have? We have 317. And how many plus plus X we have? We have 401. Now regarding double recombinant, we have 1 plus 0, 1 plus 0, but then again 2 of these happened. So 2 times the value. Again, the double recombinance between Y and X, there has been 2 crossovers. It wouldn't have been the case if we were talking about Y and W, because that would have been 1 crossover. Or between W and X, that would have been just 1 crossover. But in this situation, since Y and X are too far apart, it could have 2 crossovers, and so we multiply the value by 2. Okay, so these are the values that we got. I've already calculated the number for you. So single recombinants plus 2 times double recombinants times 100 divided by 2000, which was our total number of gametes. So here you can see that the total number of gametes adds up to, so there is 37.4 map unit distance between genes X and Y. And let me show that to you. Between gene X and Y right here, there is 37.4 map units. Now, if you would like to pause the video and as a practice try to find the distance between Y and W and then W and X, but just keep in mind, for these situations, you don't need to multiply the gametes with double crossover by 2. You just add them up. Okay, if you need to pause the video, go ahead. Here is the table. Otherwise, I can move on with answering the next question, which is what's the distance between Y and W? So first, we could have had a crossover between Y and W. And then another crossover we could have had between Y and W. And then we don't care this time if there is another crossover between W and X, because we are not trying to find the distance between W and X. So since here in this situation, only 1 crossover happened between Y and W, these double crossover gametes are only added once. We don't multiply it by 2. So the distance between Y and W is 12 plus 16 plus 0 plus 1 times 100 divided by total gives us 1.4 map unit. So again, the distance between Y and W is 1.4 map unit. So I'm going to erase this one right here. So 1.4 map unit is the distance between Y and W. Now let's do the same thing with W and X. You have to find the recombinants between W and X, add them up, and then we can also have another recombination between W and X in double recombinants. We don't multiply the number by 2 again because only one crossover happened in these double crossover gametes. So again, you add the numbers times 100 divided by total gives us 36 map unit. So going back right here, the distance between W and X is now 36 map unit. And if you add up 36 with 1.4, the total distance is 37.4 map unit. Okay, and here is redrawn distance that I've just shown you. 37.4 was the distance between Y and X. 36 was the distance between W and X, and 1.4 was the distance between Y and W. The next concept that I would like to discuss is sex-linked genes that were discovered again by Morgan. So basically what Morgan was trying to do is that he was trying to prove that Mendel was wrong. And so he started his genetic works with Drosophila, and what had happened was that one time he has noticed that one of the flies has a white-eyed color. And when he started to do experiment with that white-eyed color, that's the time that not only his findings supported Mendel's, but it also helped him to come up with the conclusion that there are some genes that are located on sex chromosome. So this is basically what had happened. Morgan has found a white-eyed fly. Okay, now he used that white-eyed fly and he crossed it to a female fly, and I've already shown you the genotype. So of course Morgan didn't know how this whole thing is happening, but later on he came to the conclusion of X-linked genes. And so since we already know that, I'm going to show you the genotype of every one of these flies. So we know that the fruit fly, the male is XY, similar to humans, and female is XX. All right. Now the color for the gene for the color of the eye in fruit fly is located on the X chromosome, and so therefore he had a mutant fly that had a mutated X gene. So here I've shown you the mutated gene as a white color right here. So this gene was mutated. Once he crossed that with the female flies that had normal genes, he noticed that the next generation, both males and females, had a red color. And the reason for that is that here, X goes with either Y or with the other X. So here we get different combinations. XX, which is heterozygous, and XY, which is red color. Again, white color here was recessive to the red color, and that's why it didn't show up in the F1 generation. So here we have the F1 generation. And if you try the other chromosome, you would still get the same thing, XY or XX, which is the recessive gene right here. Now, in the next step, he has performed a cross between F1 generations. So here you can see that the combinations we get is this X chromosome with the white-eye gene comes with a Y chromosome. So consequently, these flies will have a white-eye color. The other combination that you get is this X chromosome with the white-eye gene coming with another wild-type gene, but since red eye is dominant, it won't manifest itself. So here we still get a female with a red eye color. Now similarly, we can try to find out what combination we get from the other X chromosomes. So wild-type X chromosome coming with the Y chromosomes gives us a male with a red eye color. Or this wild-type X chromosome coming with a wild-type X chromosome, again, that gives us red eye color. So the combination of this cross has given Morgan a 3:1 ratio. But then given that the white-eye F2 flies were all males, it has led Morgan to the discovery that there are some genes that are located on the X chromosomes. So these are referred to as sex-linked genes. And the reason that X and Y are determining the sex in different species, like in humans and in flies, is that the Y chromosome has certain genes. Like for instance, in humans, it has SRY gene that leads to the development of the male sexual organs. And from there on, other sexual characteristics and phenotypic changes happen. So there are genes on the Y chromosomes that determine the path of that embryo to become male. If that gene, however, is not present, by default, humans are designed to become female. Okay, so that's why if there is Y chromosome, there would be male development. If there is not, then females will develop. Now, genes that are located on X chromosome are called sex-linked because their appearance follows also the sex of that individual. All right, now let's go over extranuclear inheritance. So there are certain organelles that have their own DNA and they replicate independent of the cell. So their genetics is not dependent on the nuclear genome. And these examples are, for instance, mitochondria and chloroplast. So going back to the process of evolution, it's thought that the modern eukaryotic cells that are able to use oxygen by their mitochondria for energy production, that they have developed by engulfing some primitive aerobic prokaryotic cells. So imagine if we had an anaerobic eukaryotic cell millions of years ago that is not able to use oxygen. But then there was some prokaryotic cells that can use aerobic oxygen to produce energy. So now this cell engulfs the aerobic prokaryotic cell, and over millions of years, this prokaryotic cell is turning into mitochondria. It still has its own genetic material. It still divides by binary fission, similar to prokaryotes. And then later on, same thing happened with plants. Plants that had mitochondria, some of them engulf photosynthetic prokaryotes that has turned into chloroplasts. Chloroplasts, similar to mitochondria, they got their own genetic material. They replicate by themselves through the binary fission process. So that's why that the replication of mitochondria in human is independent of the nuclear genetic material. Now, what happens when an egg is combining with a sperm? The egg is carrying most of the mitochondria. Okay? And so once the zygote is developing, the zygote mitochondria are primarily from the egg, which has been inherited from the mother. And that's why we inherit all our mitochondria from our mothers. And so if there is a mitochondrial disorder, that can only get transmitted from mothers, not from fathers, because we don't inherit our mitochondria from fathers. And so here I have shown you a drawing that a female contributes to the development of the mitochondria, and then both female and male contribute each to 50% of the nuclear DNA. So here, by going over extranuclear inheritance, that's what I'm referring to. Mitochondria that their genetic is independent of the nuclear DNA. Next, I'd like to go over mutations and how they lead to different genetic variability. So basically, over time, our genome can undergo mutation that changes the sequence of DNA. And this change can potentially alter the structure of proteins that those DNA are coding for it. Most of these mutations are deleterious, means that they're not good, but sometimes some of these mutations are good. There is a new protein that turns out to be very effective in helping with survival of that organisms. And so that's why that over time, these beneficial mutations can add up to come up with new function in different organisms. So mutations can develop from errors during DNA replication. So that's one mode of developing mutation. Second, mutations can develop from exposure to mutagens. And these are chemicals or compounds or stress factors that enhances developments of error in DNA. And examples of mutagens is, for instance, like X-ray can cause double breaks in DNA, or like, for instance, UV light can cause mutation in DNA. And then finally, DNA can get damaged. DNA damage by free radicals. Okay, and free radicals, a lot of them are being produced by mitochondria. So reactive oxygen species are mostly being produced by the mitochondria, and the reason for that is that mitochondria is using electron transport chain in order to transfer electrons to O2. As a consequence of which, now there is chance of developing reactive oxygen species, and these reactive oxygen species can damage DNA. Now, regarding different types of gene mutations, we've already discussed this in our DNA replication video, but here I would like to give you a brief review of different gene mutations. So silent mutations are mutations that do not really affect the coding of protein, because that mutation yields still the same amino acid. And here I'm showing you an example of how the template DNA strand is used to produce an mRNA. The other DNA strand is called a coding DNA strand because its code is the same as mRNA, except that instead of thymine, there is uracil in the mRNA strand. Okay, so the DNA template strand was used to transcribe mRNA. And now this mRNA, through the translation process, different codons will code for different amino acids that will be put together by the ribosomes. So in the first example here, we have a normal translation process and there is no mutation. However, if there is a mutation, for instance, that TTT converts to TTC, but then this codon is still coding for the same amino acid, so there is no change in the protein structure. And this would be an example of silent mutation. However, if there is a mutation that now the codon is coding for a different amino acids, then that would be an example of missense mutation. So here you can change that phenylalanine has been changed to leucine. And then nonsense mutation is when codon changes into a stop codon, which is the code that tells us, okay, that's enough. You got to stop translating at this point. So this would be an example of nonsense mutation. And going back to this table, silent, there would be the same amino acid. Missense, it would change the amino acids. And then nonsense, an early stop codon will be created. And the memory aid that I have for you here is called stop the nonsense. So nonsense mutation is due to development of a stop codon. And finally, frame shift mutation happens when there is either addition or deletion of a nucleotide. And the reason for that is that imagine if we add a new nucleotide right here. What happens? Since the genetic codes comes in triplets codons, the whole sequence will be affected because now there would be one nucleotide shift. And these amino acids will no longer be similar to what we had earlier. So usually the frame shift mutations are causing the most devastating phenotype because they completely change the amino acids that are there. So here I'm giving you an example. We had the mutation right here. Okay, and there is a base pair that is added, a G that is added. So from this point on, all the amino acids that we are getting are nothing similar to the original amino acids that we had there. So everything has been changed there. Okay, and that's an example of frame shift mutation caused by addition or deletion of nucleotides, which may result in misleading of the downstream nucleotides. Moving on to the chromosome mutation. So what I just explained so far for you was an example of gene mutation. Next, I'd like to discuss chromosome mutation. So these are basically mutations that change the structure of chromosomes, and therefore many genes will be affected. An example would be a deletion where part of the chromosome is deleted. So here you can see that D is deleted and give A, B, C, E. So part of the chromosome is deleted. Alternatively, there could be a duplication and part of the chromosome duplicates. So here in this example, BC duplicates and we have two BCs right here. Another type of chromosome mutation is called inversion, where part of the chromosome is removed and re-added in reverse orientation. And here is an example for you. BCD is now changing in the direction DCB, and that's an example of inversion. Insertion is the next type of DNA chromosome mutation, where a piece of one chromosome is removed and inserted into another chromosome. So as the name implies, insertion. And here is an example of insertion. C and D are removed from these chromosomes and are added to the yellow chromosome shown up there. And then finally, we can have translocation, where parts of one chromosome exchange with the other one. So in this example, you can see that I is exchanged with A and B. So A and B are moving to the other chromosome, and I is moving to the purple chromosome. Okay, so we are basically done with the first part of this lecture. In the next lecture, I would still like to continue on the rest of the topics we haven't covered here, which are the potential causes of genetic or phenotype anomaly, such as chromosome non-disjunction. We haven't discussed that yet. I would like to go over nucleotide mispairing, and then discuss how transcription and translation could also lead to mistake in the formation of proteins. Okay, so we would continue our lecture on those topics. Once we cover those, then we will go over analytic methods, evolution, and then finally go over some practice questions. Okay, so that concludes our first part of this lecture. Thank you all for listening, and I will see you for the second part of the genetics and evolution lecture.